Hello, good evening. Welcome to the back half of our lecture on inflammation and repair. Um, I intentionally left this part out for P1 because this can get a bit heavy, and I wanted to um, compartmentalize the cells and molecules that you were exposed to during the different phases of acute inflammation. But it is important to recognize that chronic inflammation is part and parcel of the whole process. That's point number one. And point number two, um, repair is intrinsically tied with the process of chronic inflammation. So, as we proceed with the lecture, please keep in mind these very important points. First, um, we need to differentiate acute versus chronic inflammation, and we need to identify what causes inflammation to proceed from the acute phase to the chronic phase. We need to identify what are the changes in the process, what are the changes in the intent of the body as it shifts from acute to chronic inflammation, and what happens to the tissues as we shift to the chronic phase of inflammation. Okay. Um, we will also be exposed to the other inflammatory cells, particularly the monocyte, macrophages, lymphocytes, and plasma cells, and we will contrast that to the cells of the acute inflammatory response. We will also see that there is a cross interaction between the inflammatory cells of chronic inflammation and the cytokines they produce, and how it further stimulates acute inflammation. So it creates a loop wherein the acute inflammatory cells are also stimulated. And we will also talk about the systemic manifestations of inflammation and how it relates to the patient, how these signs and symptoms manifest, and in the worst case scenario, how uncontrolled inflammation can lead to severe morbidity and even mortality for the patient. Okay? So, chronic inflammation is inflammation of prolonged duration. And unlike acute inflammation, where it basically takes place over the course of the first 48 hours, chronic inflammation takes place over a course of weeks to years. So, if the inflammation doesn't subside, if the inflammation is not controlled, chronic inflammation persists. And aside from the inflammation, there is concurrent, prolonged, long-standing issue to the injury to the tissue, all accompanied by healing. Um, However, because of the concurrent inflammatory state and attempts of healing, this is often accompanied by fibrosis or scar tissue formation because the healing is not completely organized, okay, or the healing is not completely regenerative. Okay, so there's something wrong with the tissue. Okay, there's a constant state of damage while the body is trying to heal itself or attempting to repair itself. So by virtue of that, you have two opposing um, processes going on. Chances are there will be um, a problem in how well the healing has taken place. Okay. So as mentioned, chronic inflammation is slow. Unlike acute inflammation, which takes place again in the first 48 hours, this takes place over weeks to months. And the end effect of chronic inflammation is fibrosis or collagen deposition or scar tissue formation. Okay? Now, this may occur in two specific patterns. The first pattern is chronic non-specific inflammation. So you have a host of um, chronic inflammatory cells, lymphocytes, plasma cells, or macrophages. And you also have granulomatous inflammation, where you have macrophages, or the histiocytes, forming granulomas. This is particularly true in certain infections and autoimmune diseases like TB, sarcoidosis, leprosy, um, etc., etc. Okay, so in granulomatous inflammation, you not just have the chronic inflammatory cells, but you also have a predominance of um, a component of the monocyte macrophage system, particularly histiocytes, which gain more cytoplasm as they react to the long-standing infection or long-standing damage to form granulomas. Um, as you might have recalled, a granuloma is a collection of epithelioid histiocytes rimmed by chronic inflammatory cells and fibroblasts. It serves to contain the cause of the chronic inflammation. Now, when does chronic inflammation arise? One of the most common reasons behind chronic inflammation are infections that the body cannot get rid of. Now, usually, um, the body is fairly effective in trying to curb these infections. However, there really are infections um, or there really are microorganisms that have their own means to escape the immune system. And this is particularly true, number one, of the Mycobacteria family. The organism that causes tuberculosis, Mycobacterium TB, the organism that causes leprosy, Mycobacterium leprae, okay? So, and other microorganisms like Treponema pallidum, which causes syphilis, okay? Now, we know in the first 2 days of infection, of course, you're going to have the acute inflammation and acute inflammatory stage, fever, um, the cardinal signs of inflammation. And in the acute phase of inflammation, the body is trying to kill the organism. Okay. Now, there are organisms, as mentioned, that cannot be rid of using the um, using the um, mechanisms employed by the body in acute inflammation: hypochlorite secretion, free radical formation, phagocytosis. Hindi, dili makalaban. Okay? So the body tries to contain it instead. The body tries to prevent it from spreading. So it forms granulomas. And this is particularly true for these organisms. Okay? Treponema pallidum, I'm sure you've heard of TB, I'm sure you've heard of leprosy. Treponema pallidum is a sexually transmitted infection. It is venereal. It enters the skin. It enters the genitals, and this usually manifests as a painless sore in the penis. And this painless sore is known as a chancre. 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 Okay. After a week or two, after several days, the chancre disappears, and um, there is going to be disseminated syphilis. The syphilis is going to course through the bloodstream. At this time, the body will react by presenting with rashes. And the rashes of Treponema pallidum is peculiar in that it will involve the hands and feet, the palms of the hands and the soles of the feet. And that manifestation is quite rare with all other entities that cause rashes. Okay, so that's one thing that's peculiar about syphilis. And over time, over the course of several years, syphilis um, will go to the brain, and this will manifest as neurosyphilis. Okay, loss of balance. It's known as tabes dorsalis. Okay, aresthesias, um, a tingling effect. Okay, and ultimately the patient becomes demented. I'm sure you've seen movies portraying Victorian England. Um, they actually have sanitariums before for women who go crazy, and this is because of the high rates of prostitution during that time. Okay, and they didn't know na syphilis actually causes damage to the brain over time. So it has been said na the reason why there were a lot of crazy women during Victorian England because of the high rates of prostitution. These were cases of neurosyphilis. Okay. Now, all regardless of whatever the cause, um, they tend to establish persistent infections, and the end result is a reaction that calls out T lymphocytes. Okay, and this response is known as delayed type hypersensitivity. Okay, Now, it's not just infections. It's not just infections that can cause chronic inflammation. You can also have inflammatory diseases, particularly autoimmune diseases. Autoimmune diseases are diseases wherein the immune system remains uncontrolled. The immune system starts to attack different parts of the body. Okay, so for example, um, rheumatoid arthritis uh, has antibodies directed towards the joints. Inflammatory bowel disease affects the colon. Okay, psoriasis involves the body mounting an immune response against certain layers of the skin. Okay, so that's an autoimmune disease. That's the principle behind autoimmune disease. Or you can have allergic reactions to um, antigens or to substances that are present in the environment. Okay, so you can also have inflammatory diseases there. Okay, other causes include prolonged exposure to potentially toxic agents. So, for example, you have dust particulates. Okay, dust particulates. This is particularly true for industry workers. Okay, if you work in mines, for example, if you work in factories, like those that produce plastics, silicates, okay? These are very small and they can enter the spaces of the lungs. And over time, the body will recognize these silica as foreign. So the body will try to get rid of it, okay? And it mounts a chronic inflammatory reaction, okay? Now, the last setting by which chronic inflammation may arise is through certain long-standing disorders, particularly in Alzheimer's disease, wherein um, uh, short-term memory is affected. In Alzheimer's disease, this is in vernacular, ango-ango, di ba? In Alzheimer's disease, this is a neurodegenerative disorder wherein what is impaired is the short-term memory formation. This is the reason why kung mga tao nga ango-ango, usually they will recount um, mga memories na karaan pero dili sila makakuan unsay nahitabo recently or last week. Okay? Okay? Now, chronic inflammation is characterized by three overlapping processes. Okay? Three overlapping processes. First, you have the infiltration with the chronic inflammatory cells. And the chronic inflammatory cells are known as the mononuclear cells. Okay? The first group are the macrophages, which are essentially specialized monocytes. They are monocytes that have reached the tissues. Okay? And then you have lymphocytes, which are composed of T and B lymphocytes. And you have the mature antibody producing B lymphocytes known as plasma cells. Okay? Now, because you have these inflammatory cells, like acute inflammation, there is also some degree of tissue destruction. However, unlike sa acute inflammation, na the damage to the tissue is very strong, it's very destructive, but it usually happens within a shorter course of time. In chronic inflammation, the damage is indolent. It's not as strong, but it persists for a very long time. Okay? So, aside from the inflammation, you also have tissue destruction, but at the same time, since it's happening over a prolonged period of time, while the damage is going on, the body is also working to try to repair itself. Okay? And the body tries to repair itself through two means. One is the formation of new blood vessels, which are leaky and immature, and this is known as angiogenesis, and the deposition of collagen, and this is known as fibrosis. Okay? Now, in contrast, acute inflammation, if you remember, is characterized by vascular change, transient vasoconstriction, then vasodilation, and then exudation of plasma leading to edema, and the mobilization of neutrophils. That's the difference sa dalawa. Okay? So this is an example of chronic inflammation. Okay? Here, sa arrows, you will see mga chronic inflammatory cells. Okay? And then here, sa asterisk, you will see lymphoid. You see lymphoid aggregates. Okay? And in the interstices, letter B is actually, it's not very clear in this picture, but I think this is acute inflammation. You have a lot of neutrophils. This is pneumonia. Okay? And you have congestion of the blood vessels. Okay? So this is acute inflammation. Now, cells in chronic inflammation, again, mas marami siya. And the interactions are more complex. This is, again, the reason why I decided to separate this topic after P1. Kaya daghan na kaayo ni siyag mga bagong cells, bagong chemical mediators. Okay? And again, this is mediated by macrophages, which are tissue monocytes, and lymphocytes and plasma cells. Okay? So, tissue monocytes, again, are known as macrophages, and depending on where they are, they tend to be given specialized names. Okay? So, in the tissue, again, they are known as macrophages. In the blood, they are known as monocytes. Okay? Kung sa liver, these macrophages are known as Kupffer cells. In the spleen and the lymph nodes, they are known as sinus histiocytes. In the CNS, they are known as microglial cells. And in the lungs, they are known as alveolar macrophages. In addition, in the skin, they are known as Langerhans cells. Not to be confused with Langhans giant cells. Okay? So again, this is a diagram showing us the mononuclear macrophage phagocyte system. Okay, we know that the precursors of the cells circulating in the blood all come from the bone marrow. Immature precursors of monocytes are known as monoblasts. Okay, after 48 hours of inflammation, after the acute phase, cytokines are secreted, which causes the release of these monoblasts from the bone marrow into the circulation. And they will circulate, and depending on chemical stimulation, cytokine stimulation, they go to where the inflammation is, particularly in the tissues, in which case it becomes enlarged, it becomes bigger, it acquires more cytoplasm, and at which point it is now known as a macrophage or a macrophage. Okay? Now, if the macrophage responds to an inflammatory process, okay, it is said to be activated. And as it becomes activated, they are known as activated macrophages. And that's what I keep on mentioning. Activated macrophages tend to acquire more cytoplasm. Okay, they tend to acquire more cytoplasm. And because it acquires more cytoplasm, it tends to look like epithelial cells. Mura siya epithelial cells, because epithelial cells are characterized by large amounts of cytoplasm. That's why we call them epithelioid macrophages. Macrophages that look like epithelial cells. Okay? And over time, as soon as the more that they are stimulated, these epithelioid cells tend to fuse together, and they become known as giant cells. Okay? Now, another pathway that through which macrophages can take is a process known as differentiation. They become specialized macrophages of the organs where they go. In this case, they become microglia, Kupffer cells, depending on where they are in the body. Okay. Now, macrophages may be activated by many stimuli. Okay, so medyo confusing siya. You don't really need to identify specifically what the interleukins are, but suffice to say that there are many ways by which macrophages are activated. Okay, and there are actually two pathways which we'll talk about later, and these are the classical and the alternative pathways. Okay, now they can be activated by cytokines, and this is part and the most important cytokine that activates macrophages is called interferon gamma. Okay, interferon gamma. Interferon gamma activates monocytes to become macrophages. Okay, bacterial products can also cause activation. Okay, now what? Why do macrophages have to be activated? Okay, now number one, macrophages become activated, and by doing so, they become more equipped to eliminate whatever invading substance there is. At the same time, aside from being able to secrete more substances that can eliminate, um, they can also secrete cytokines that start the process of repair. So it is at this stage you will see na it is actually the macrophage that is responsible both for the destruction and the attempts to repair. Okay? It will also secrete mediators of inflammation, which will orchestrate the immune system. Okay? And then it also participates in a process called antigen presentation to T lymphocytes. And we will be talking about that later. But the end result of antigen presentation to the T lymphocytes is that the T lymphocytes will secrete more cytokines, further augmenting the chronic inflammation. So, murag strengthen niya, mag-feedback loop siya. Kaya mo-present siya inflammation sa T lymphocytes, mo-present siya og antigen sa T lymphocytes produce og cytokines na magtawag og bag-o nga monocytes to become macrophages. Okay? So it is this feedback loop that is responsible for the long-standing injury seen in the cells. Okay? Now, in chronic inflammation, there is macrophage accumulation by calling in more monocytes from the circulation, and this is mediated by the cytokines, the additional cytokines that are produced. So mag-feedback siya. Local proliferation of macrophages. So macrophages will undergo cell division in the tissues, and then immobilization of macrophages, which means that the macrophages do not get out of the site of inflammation. Now, if you have activated macrophages, as I've mentioned, diba, macrophages gain more cytoplasm and they become known as epithelioid histiocytes. Once you have a collection of epithelioid histiocytes ribbed by chronic inflammatory cells like lymphocytes and plasma cells, and fibroblasts which secrete collagen around this collection of activated macrophages, you call this a granuloma. Okay, so that's the macrophage. Now, moving on to the other component of chronic inflammation, Now, moving on to the other cells that are involved in chronic inflammation, you have lymphocytes, and lymphocytes are the smallest WBCs, and you have two subtypes. You have T and B lymphocytes. Okay. Now, B lymphocytes mature to develop into plasma cells. Okay, and how do plasma cells look like? Think of it as a lymphocyte that has acquired more cytoplasm, mas naase cytoplasm, and the nucleus tends to be eccentric. It's off to the side, and it's very circular. Okay, and sometimes when plasma cells start to secrete antibodies, they tend to have this collection of antibody molecules within the cytoplasm. Okay, those are plasma cells, and their job is to secrete antibodies. T lymphocytes, on the other hand, secrete not antibodies, but instead secrete cytokines, essentially the molecules that orchestrate the immune system. Okay? CD4 positive T lymphocytes promote inflammation and influence the nature of the inflammatory reaction. So the CD4 T cells, CD4 positive T cells, serve as the maestro of the immune system by secreting the cytokines. And the cytokines tend to stimulate different parts of the immune reaction. This is the reason why HIV causes immunodeficiency, because it kills the cells that orchestrate the immune response. Okay. Now, at this point, I'd like to talk about cell-mediated injury. Again, cell-mediated injury is mediated by T cells. Okay, it's mediated by T cells in contrast to antibody-mediated immunity, which are mediated by B cells and plasma cells. Again, cell-mediated T cells, antibody-mediated or humoral-mediated immunity, that's B cells or plasma cells. Okay, now at this point, let's talk about cell-mediated immunity. Okay, and at this point, let's talk about the different types of T cells. Essentially, let's talk about three. We have dendritic cells, CD4 positive T cells, and CD8 positive T cells. Okay, now CD8 positive T cells are also known as cytotoxic T lymphocytes or cytotoxic T cells, CTLs. And its function is simple. It destroys cells that are infected by intracellular organisms, like intracellular bacteria, viruses. When the infectious agent is inside the cell, at yun siya using CD8 positive T cells. Okay? Now, dendritic T cells serve to gobble up. They are phagocytes. Dendritic cells tend to gobble up. They tend to eat the organisms and bring them to the lymph nodes. They bring them to the sinuses of the lymph nodes, or the spaces between the follicles of the lymph nodes. And once it has gobbled them up, it processes these organisms na gikaon niya and selects specific portions of the organisms. Okay? It selects specific portions of the organism and it will process it inside the cytoplasm of the dendritic cell, and the processed portion of whatever it ate up, dadun sa surface. It will express it in the surface. Okay? And that is a process known as antigen presentation. Okay? It's the dendritic cell who does that. And this is known as antigen presentation. A dendritic cell is responsible for antigen presentation. Hence, it is also known as an APC, antigen presenting cell. Okay? Now, CD4 lymphocytes serve to interact with dendritic cells that express antigens on their surface. And once mag-react ang CD4 positive T cell together with the dendritic cell with its processed antigen, the CD4 positive T cell will secrete cytokines. Okay? So that is essentially cell-mediated immunity. Okay? Now, you have different subtypes of helper T cells. Okay? There are helper 1 T cells. Okay? So these are CD4 positive T cells or helper T cells. You have 3 subtypes, helper 1, helper 2, and helper 17. Helper 1 serves to secrete cytokines that serve to stimulate macrophages, helper 1. And the major cytokine here is interferon gamma. Okay? Helper 2 serves to are stimulated by interleukin 4, 5, and 13, and they are involved in allergic reactions and parasitic reactions. Okay? And you also have interleukin 17 and interleukin 22, which stimulates helper 17 T cells, which serve to recruit neutrophils and monocytes. And this is particularly useful in certain autoimmune diseases. Okay? And then, this is actually MHC. Now, we're going to talk about natural killer cells. And natural killer cells are very similar in that they are also associated with MHCs, or major histocompatibility complexes. Now, remember what I told you about CD4 positive T cells and antigen presenting cells? Now, antigen presenting cells, as mentioned, gobble them up, gobble substances up, or gobble microbes up, and then they process the antigens of whatever organism they gobbled up and complex them to small molecules known as MHC, major histocompatibility complex molecules. Okay, and that is what they present to the surface. Okay, now the MHC molecules seen in antigen presenting cells are known as MHC class II molecules, and they react with CD4 positive cells. Okay, however, um, However, you also have another type of MHC molecule, and these are type 1 molecules. Unlike MHC class II molecules, which are found only in antigen presenting cells, MHC class 1 molecules are found in all cells. Okay? They are found in all cells which have nuclei. Okay? Now, normally, these MHC class 1 molecules are recognized by receptors in the natural killer cells, and they are said to be inhibitory receptors. And because these are inhibitory, NK cells are not activated. Now, in infected cells, or cells that are have undergone mutations, um, the inhibitory receptors are reduced. Okay, so the NK cell will not be able to recognize the inhibitory MHC1 molecules in the surface. So there is nothing inhibiting the reaction. Thus, the NK cell will try to destroy those infected cells or tumor cells. And this is known as NK-mediated killing. And this is particularly useful as defense for tumor cells. Okay?
Now, let's talk about the second branch of immunity where the lymphocytes are involved. The first one, as mentioned, was cell-mediated immunity mediated by T cells. For this portion of the lecture, we'll be talking about humoral immunity, and this is mediated by lymphocytes, B lymphocytes. Okay? Now, B lymphocytes, as mentioned, react by producing antigens. By producing antibodies. Okay? Now, we need to be familiar with the concept of naivete of B cells. Okay? Naive B cells are B cells that have not yet been exposed to antigens. Wa pa siya mga virgin B cells, wa pa siya exposed to an antigen. Okay? Now, if they are exposed to antigens, the B cells either divide or either proliferate, or they differentiate into plasma cells. And the plasma cells serve to secrete the antibodies. Okay? Now, some of these plasma cells undergo heavy chain class switching to produce the different types of antibodies. So first, it's IgM, but over time, they produce IgG as well. Okay? IgM is very potent, but it does not last long. Okay? IgG is abundantly secreted, but it takes time for them to be secreted. Okay? And you also have specialized immunoglobulins like IgA, which is the major antibody found in mucosal surfaces, sa mouth, sa nasal cavity, and IgE, which serves to stimulate mast cells and facilitate the granulation of mast cells to produce histamine. Okay? Now, let's take a look at this diagram. Okay? So, at this part, you have a naive B cell. So, kung naive B cell, wala pa siya na-expose. Okay? Wala pa siya na-expose. Now, at this point, it is important to recognize that kung naive B cell siya, In general, sorry. In general, um, there is one type of lymphocyte to produce one type of antigen, to react with one type of antigen, and it will only produce one type of antibody. Okay, let me explain that in more concrete terms. Let's say you have B lymphocyte A. B lymphocyte A has on its surface antibody A, and it can only produce antibody A on its surface. Antibody A na gid siya. And because there is specificity of antigen and antibody, it will only react with antigen A or organism A. Okay? Now, if organism A will react with the antibody A on the surface of lymphocyte A, okay? So, nag-react na sila. A reacting with A. Okay? Very, very specific. Okay? Sila ra gyud pwede ang mag-react. Okay? Now, there are so many antigens in the known universe. There are so many antigens a human body or a cell can be exposed to. It's infinite, diba? So, if that is the case, that there is only one lymphocyte capable of producing one antibody, it goes on to it can be said that it does not make sense to have multiple copies of one lymphocyte. Kaya daghan kung posibleng antigen. Diba? So what's going to happen is, instead, to save space sa mga lymphoid organs nimo, if ma-expose ang lymphocyte A to antigen A, ang mahitabo niya is mo proliferate ang imuhang lymphocyte A to produce multiple copies to fight organism A in the body. So managhan siya. And this is the reason why it takes time for the antibodies to be produced. Diba na kay mga booster? Diba? It takes time. The first contact between the microbe and the lymphocyte is known as sensitization. And during the sensitization phase, managhan pa siya to try to replicate itself para ma-counteract ang organism. Daghan sila magtabang. Some of these lymphocytes will differentiate to become plasma cells, which will produce the antibodies. And ang antibody nga iyang gi-produce is the same antibody nga gi-express sa iyang surface. It's just that isikrit na niya tanan to try to neutralize the infection. Okay? And these antibody-secreting lymphocytes are known as plasma cells. Okay? However, naay uban lymphocytes na during proliferation dili mo mature into plasma cells. Instead, they just remain lymphocytes and hide elsewhere in the body. And these become known as memory B cells. Okay? They become known as memory B cells. Katong plasma cells mag-secrete og antibody, they will neutralize, they will act as opsonins, and they will facilitate lysis of the organisms. They will facilitate complement activation. However, there is what is known as class switching. So IgM ma-convert to IgG, okay, etc., etc. Now, plasma cells, as mentioned, are mature B cells. They serve to secrete antibodies, and antibodies serve to neutralize. They serve to activate complement, especially IgG. They also serve to act as opsonins, particularly IgG. Okay? Eosinophils are involved in both acute and chronic, and they are mediated by IgE. We see them in allergies and parasitic infections. Now, they are known as eosinophils because they contain red granules. Eosino is red-orange. And the major component of the granules of the eosinophils are MBPs, or major basic proteins, which are toxic to parasites. Okay? And then another type involved in chronic inflammation are mast cells. Okay, they express on their surface IgE, And then IgE, if it reacts with an allergen, will stimulate degranulation and release of histamine. And we know that histamine is a very potent vasodilator. And this is also the reason why if you have massive degranulation of mast cells, when there is extreme allergic reaction, you will have anaphylactic shock because mag-vasodilation, mawagtang ang blood pressure, mubagsak ang blood pressure. Okay, this is known as anaphylactic shock. Okay, these are systemic effects of inflammation. So essentially, this is mediated by the cytokines that are produced during the inflammatory phase, and the substances that are produced during the inflammation as a group, we call them acute phase reactants. Okay, so IL-1 and TNF generally causes fever, loss of appetite, and the general feeling of tiredness during the sickness phase. In the bone marrow, it also stimulates to mobilize the white blood cells. Okay, you have they can also act as endogenous pyrogens, but bacterial products may also facilitate the production of fever. Okay, now IL-1 and TNF or IL-6 seen by gram-negative bacteria will act on the temperature regulating portion of the brain, particularly, and this is known as the hypothalamus. Okay, and this will facilitate the secretion of prostaglandins. Okay, it stimulates the sympathetic nervous system, which causes vasoconstriction. Okay, and because of that, heat is dissipated, and that's why you give off heat. Okay, so that's a principle behind fever. Okay, the reason why you have a decrease in temperature during fever when you take in anti-inflammatory agents is because they act on the prostaglandins that are produced during the inflammatory phase, anti-inflammatory. So, i-decrease na yung temperature. Okay? And this is one effect, of course, leukocytosis. We have a lot of neutrophils. Okay? You have massive leukocytosis. Sometimes, grabe ang infection that sometimes mga more immature forms of the neutrophils are shifted out of the bone marrow, and this is known as left shifting. Okay? Here, this is a stab cell. As you can see, dili pa kayo siya multilobated and like these neutrophils. Okay? Here, mura siya band. This is known as a stab cell or a band cell. This is a mature neutrophil. And this is known as left shifting, the release of immature WBCs from the bone marrow due to massive infection. Okay? Now, the massive release of inflammatory chemicals or cytokines causes blood to become sticky. Okay? And this leads RBCs to fall slowly if you let it stand in a column or in a tube, because viscous ang plasma, muhinay ang bagsak sa RBCs, and this is manifested by RBCs taking more time to sink to the bottom of the tube, and this is known as erythrocyte sedimentation rate, and this is a test that we use in the lab to detect inflammation, ESR. Okay, and this is the setup. You just let it stand for 1 hour or 1 to 2 hours depending sa ano. And then you measure the fall. Okay? So if taas ang ESR niya, that means it took time for blood to go down. So that means na active inflammation taking place. So as a summary, again, chronic inflammation is a distinct entity, different causes, different chemicals and cells involved, and different reaction to tissue. Okay, it involves a lot of cell types, including lymphocytes, macrophages, plasma cells. And this will manifest as systemic signs and symptoms, and these are mediated by cytokines. Okay, so thank you very much. This has been a pretty long lecture. I'll see you sometime this week for a live feedback session on this topic, right? Thank you.